So good morning, ladies and gentlemen. I will present here Department of Cognitive Systems and Neuroscience and Department of Biomedical Engineering and Assistive Technologies. My name is Lenka Vysloužilová and I'm head of one of these departments. On the left side, you see the list of our workers. We are quite bigger than last year, 2024. So our group is now about 20 workers, and Department of Biomedical Engineering is about 3 workers. So our results, we have 8 quartile 1 articles, means 4 in decimal 1 and 4 in quartile 1. You can see on the board names of articles. We have six conference papers, and docent Lhotská has invited talk on World Congress. Our groups have 3 international projects. The biggest one is Clara, where we participate with other groups, and our leader is Václav Zeman, and other projects are mainly Czechs and mainly from health domain. So that's our group. And I'm the first speaker. I would like to present here our article named Smart Alert Machine Learning Based Patient-Ventilator Asynchrony Detection System in Intensive Care Units. This article is part of VentConnect project. VentConnect project starts in 2020 as one of COVID-19 projects. Yeah, it's really 6 years now. We do it with cooperation with Department of Anesthesia and Intensive Care Medicine, Královské Vinohrady University Hospital. And our goal was monitoring of ventilators and vital signs from patients and send it to remote doctor room because pandemic. So here you can see the view of screen of ventilator. There are two pairs. One is flow. The upper is called flow. The down is volume, and our goal is analyze these curves and identify the risk of ventilation. Now we have 400,000 videos of these screens, and after cleaning and filtering, we have around 200 thought videos recording from one type of ventilator. So this is our starting data to learning. here's how whole system looks like. We have bedside, means side with patient. There is lung ventilator and monitor vital side, and we take data from this monitors to our unit, vent unit, and we anonymize data and send it to server. It's the middle box, and after classification and so on, we send data to client. It's in our case a mobile phone which has the doctor, and or we send data to clinical system in this case MetaVision. So quite complex. So how we set the learning. We have one normal alarm level, means 0 and 3 rises, and 3 is the worst. So we set our learning to specificity 1.999 to this worst level. So we know that every worst level are detected. So pipeline, we have two phases. One is learning, one is recognition, classic machine learning. We take photos or screenshot from ventilator. We find the image of the curves and find the values of points on these curves. Because there are 200,000 of time series, it's not possible for doctor to annotate this huge data set. We take we do the cluster analysis. So we change 200,000 data screens to clusters. We have 400 clusters, around 400 clusters, and the doctor annotate these clusters and say if it's risky or not. So they say us the level of risk, and we have some machine learning algorithm and classic classificator, and this model say which level it is, and we after send some annotation, some notification to doctor. So here's our neural network, which is quite complex because we have curves, and these curves are different length and scale. So we must masking them and use something to the time series. On the left, you see that we add mode of ventilator and some other parameters from ventilation, and use classic neural network to find alarm level. And we have one other output which say if in ventilation is some asynchrony which can damage the patient. Conclusion: the system is deployed in clinical practice at the hospital and is currently under validation. We will analyze data from the clinical study complete last year, and we hope this will make us better prepared to next potential health crisis. Thank you.
Right. Good morning, everyone. I'll talk about something a bit different, but also biological. Okay, so my name is Němý. I'm a postdoctoral researcher at the COGSYS department. And today, in these few minutes, I'd like to talk about work in my group. We specialize in long wide matter connections, dominating using MRI or MRI. And now today I'm going to show you our most promising work from last year, which is combination of this and also cerebrovascular health. So this is very understudied field, and we took step forward here. This is the article I'm going to talk about. It's basically a part of our research line, so it's not isolated paper. It was published in NPJ Parkinson disease. So you just see we've got more methodological papers as well. For example, a neuroimage on this topic, more biological ones. We published the review as well. Then we've got also biological one. And this year I'm going to just present one paper, but this we published 3 papers on the very same topic. So this is one from stroke and another one from brain communications, but I'm just going to present the one. Okay, so the first part A here, are really the wide matter connections. And for this, we have this diffusion MRI, which is a special sequence from the MRI machine. And for this, we have radiologists in our team. And also we need some biological basis. For this, we got also medical doctors among us. And after some quite complex diffusion modeling, we arrive at these models of the pathways, like specific parts of the brain which are important, and it's important to quantify and which we can do now. And we specialize in the ones which are quite difficult to delineate like this. All right. So the cohort in question is dementia with Lewy bodies. The brains of these patients, they've got this misfolded protein alpha-synuclein, and it's basically in these spots which are called Lewy bodies. They can be seen upon MRI, for example. And so this is quite underdiagnosed disease, as it's quite difficult to diagnose and can be confused with other diseases. Brain diseases, but it's estimated that it accounts for about 10 to 15% of all dementia cases, which makes it the second most prevalent after Alzheimer's disease. Of course, we know that some of the brain pathways or brain systems are more prone to be affected by this disease than others. And just so you know, so you can see the symptoms here on the screen and can see that they are quite common to other diseases such as Alzheimer's disease or Parkinson's disease, such as hallucination or maybe disturbed sleep pattern and so on. The unfortunate thing is that there's no cure. There's no even like treatment which would slow this down. Okay, the second part, which is quite new here, is the combination of the pathways and cerebrovascular disease, because this is a very common co-pathology. You can see in these patients that they've got these pathways in this Lewy body disease quite disturbed. But we also see in these patients, older patients, that there's lots of mini strokes or larger strokes in their brains. And this is not investigated very much, but we think this can really influence the whole analysis. So we want to include it. For this reason, we again have MRI images which you can see on the screen, for example. These are like different slices of the same brain, kind of like a different height, like different height of the brain. And you can see these white spots. So these white spots are called white matter signal abnormalities of their cerebrovascular origin. And when we segment them and evaluate their volume, their volumetry, or maybe like do some kind of shock analysis, we can actually quantify how severe this is. The thing is that we know that there's a much higher amount of these lesions in these patients than in controls in this specific pathways, which we are investigating. So far, we didn't know if it is because there is much more lesions in the whole brain in general in these patients, or because this system is somehow special. For this reason, we ran an analysis, and you can see the cohort is not so large. So we got only 33 patients, but this is quite common in this kind of cohorts. And that's why we also joined the DLB consortium who get this also different hospitals across Europe. One part of this is, for example, looking at the volumetry of the lesions, and you can see that patients, of course, take up much more of these lesions in different brain lobes and in different systems. But what's interesting is that when you look at the proportion in the cholinergic system, that's the one in green, and also in the frontal lobe, are much higher than it would be expected. So these are really the regions which are out of scale, which are prone to the cerebrovascular disease. And that's really a very interesting and very important conclusion for basic science behind this, that we really need to look at these regions, not the whole brain, because otherwise the effect can be cancelled out, and that we should consider selective vulnerability. Of course, we are working on more studies now. For example, the one on the left side, we feel quite privileged that we can use this cohort. This is a post-mortem cohort, really post-mortem data, patients being scanned after 1 or 2 hours after that they've got really the MRI scanners in the nursing home, and so they can really do this, and so we can really confirm also with autopsy the diagnosis, which is quite difficult otherwise to do. And so as I told you, we published 3 papers on this topic this year. I now presented part of the one which is in red. I presented this at 2 international conferences. At the one which is called Alzheimer's and Parkinson's disease, I received a junior faculty award. And also this is part of the junior SCAR grant, which I got last year. And I was able to assemble a small team, which I now lead, and also which I would like also to thank for their work. And also we were able to get a new PhD student, Matteo Agresti, who will be with us for the next 4 years. That's it. Thank you for your attention.
Questions? Good morning, everybody. My name is Doležal, and I will present a paper with a rather complicated names that deals with combining intracranial EEG and eye tracking recordings. This work is about creation of the dataset that was published as open access in Scientific Data to provide access of this unique dataset to other researchers. This was done with collaboration with SAMT and University Hospital in Brno, Dansk University of Technology, Biomedical University, and Mayo Clinic. First, I would like to mention that the recording of the dataset relies on our custom-made eye tracking solution. This is a ongoing long-term development that was used in several projects, some of which I presented at last meetings. The solution consists of the hardware, software, and the processing pipeline. In this research, we specifically focus on pupil size, because pupil in the eye does not serve just as an aperture to control the amount of light flowing in. It also changes with emotions, arousal, cognitive effort, and so on. So for us, it's sort of a window to the brain. The recording uses a neurophysiological task battery, which is implemented directly into the eye tracking software. There are many tasks, but for the sake of time, I will present only one task, which deals with human memory. The task is called free recall task. It has several phases. In the encoding phase, 12 words are presented one by one to the subject, as you can see on the video. This is what the subject sees. During this phase, the subject tries to encode these words into his memory to remember them. Then there is a short distraction phase with simple math, so the subject cannot just repeat the words in his mind. And finally, there's a recall phase when the subject tries to recall the words from the memory and say them aloud. This is repeated in 15 trials with different words and administered with the other task before, during, and after the brain surgery. Now, why to use eye tracking in memory research? In our previous work, which was done on healthy subject and with eye tracking measures only, we have shown that pupil size reflects successful word encoding and recall. That means during the encoding, the pupil changes associated with the words differ significantly between the words that are later recalled and the words that are later forgotten. This is important. So the next step to investigate this deeper was to combine this with intracranial EEG, that means open brain, implanted electrodes. The subject undergoes the task and undergoes all the tasks during the surgery, and everything is recorded. The pupil size provides a non-invasive biomarker of memory performance, while the EEG gain direct access to the underlying brain activity. This was also published as a dataset in Scientific Data. In the presented paper, we record 23 epilepsy patients with the same task battery. There is no direct connection between our task and the epilepsy. We just use the opportunity that the brain is open so we can do this research. So there is a stimulation computer which provides the stimulation to the subject. There's the eye tracking software which records eye tracking, records audio, and also emit task events. So every anything happens on the screen is sent as events by TTL pulses to the EEG machine, so this could be synchronized. This is later combined with CT and MRI scans to provide precise localization in the brain. This is this paper. Now, this is how it looks during the surgery. We can see the screen with the eye tracker, which is providing the tasks for the subject in the red circle. Then there is the patient, which is lying in the bed and doing the task, and the surgery takes place behind the curtain. So this is view from the other side. We can see there on the right bottom the open brain with the attached electrodes. Here I would like to say that the use of our custom solution gave us greater control and flexibility in these environments where are spatial constraints. That means that we can change camera angles to accommodate camera lens to accommodate for different viewing distances and angles. There is one more picture. We can see on the left side the computer that provides the stimulation and the eye tracking, and on the right side we can see the live view from the eye tracking camera. We can see the subject. So we can also see the fixation frame that holds the subject's head in place during the surgery. And below we can see the detections, which are the pupil, and we're interested in the pupil size changes. So here is some results from the new data set. All our pupil curves in time. In the upper picture we can see that the pupil curves are modulated in different kind of experiment. In the left top view, we can see that we can even see individual responses to the words being presented. There are 12 words presented that are marked as the gray lines. In the picture below, we can see important thing. Again, we showed that the pupil curves differ significantly between the words that are later forgotten and later recalled. This means that the pupil changes carry predictive information about the subsequent memory performance. Okay, in this slide, I show causality between EEG features. This is called high frequency oscillations and Euchre area. just before the words is recalled and vocalized, we see a coordinated increase in these high-frequency oscillations as well as in increase in the pupil size. And here I provide a direct comparison of predictive power of EEG features and pupil. Both pupil dynamics and spectral error features show distinct temporal profiles for good and poor memory performance. And interestingly, in this experiment, we achieve comparable prediction accuracy for both pupil changes and EEG power in band features. But the pupil changes in the eye tracking is generally much easier to acquire than using electrodes in the brain. So in contribution, we have created a unique data set with intracranial EEG and eye tracking, which we published to give access to other researchers. We can make some other research from the brain signals. So this is an ongoing effort. So we are continuing recording with the development solution at the place it at my clinic in Gdańsk University and faculty in Nemocnice u svaté Anny. So beyond the dataset itself, we show that pupil dynamics can act as a practical non-invasive proxy or memory-related neural activity. So this is all from my side. Thank you for your attention.
Yeah, few minutes behind the schedule, so it is cut.
Good morning, ladies and gentlemen. My name is Lenka Lhotská, and I would like to present one of the results of our joint Czech-Taiwanese project. As you can see, it was focused on usability evaluation. This project was running for almost 4 years, and the main tasks there were analysis of motion, gamification, gait analysis, and usability study. Why? Because one of the main problems of current society is that it is aging, and we all know that with aging, there is decrease in physical activity, and when the activity is decreasing, then also the muscles degrade, and there are more accidents and so on. So one of the aims was to develop some devices and games that would motivate for more physical activity after injury, after some operations and so on, to bring people again in good condition. We started in this process with knee orthesis and a game supporting knee exercise both in standing or sitting position. The main sensors used were surface electromyography and accelerometers and gyrometer. After that, that was the first phase of the project. Then we focused on also very important part of our body. Sometimes when everything is working well, we don't realize how important the hand grip is and the fine motorics in small joints and muscles. But with the age, again, these abilities degrade a little bit, and till now there is no much research about it, especially about the training of these abilities, also about collection of data from the muscles and joints. So this was another part of the development. However, it was also necessary to find out how acceptable these devices will be and how feasible the data collection will be with all the population. So we used, numbers are missing, sorry. It was the translation of this presentation. We designed a study which was performed in our partner university in Tainan National Cheng Kung University and its hospital, because we had a medical doctor from there. So there were 9 older males and 8 females who participated in this experiment. All of them passed the 2 lines of the experiment, one with the traditional manual exercise and the second one with this game exercise with the sensors. And in both cases, they also evaluated their subjective feeling from the exercise and also with the feedback from the game. So the main results in this case were the usability design of the usability scale and its evaluation. And also we used the 10 items, as you can see on the slide, that they had to assess. And finally, it showed that use of this game feedback can really improve even willingness to use such a device for this exercise. It lowered stress, inconsistency, and it increased the confidence in one's own abilities. It was also interesting. There was a short or small divide between males and females, but well, we have to continue with these tests and experiments. So what to say in the end, that really also the older generation is willing to use technology, and what it brings, it's actually objective data for assessment of the rehabilitation process, and also it engages these people more. Last but not least, we plan to install this result in our smart home lab immediately when everything is settled there. Thank you very much for your attention.